Alright, this is Big Picture Anatomy with a look at the details of a motor neuron in highly schematic fashion and how a resting potential is established in that motor neuron along its axon, leaving it ready to carry a message or a stimulus whenever it is triggered or activated. So for starters, we have the cell body of the neuron, which contains the nucleus, a relatively large cell with these pieces sticking off that are called dendrites, which are the inputs for to trigger a stimulus response or an action potential down the axon of the neuron. Remember, this is a resting neuron, so there's no action potential or stimulus happening at the moment. Along the axon, if we look at it carefully, we see that it's surrounded by these hot dog bun-like Schwann cells. If we take a cross-section across here and look at that closer up, this is what it would look like. We've got the axon itself, which is actually just a tube of cell membrane, so this would be a bilipid cell membrane around here, and it's surrounded by that myelin sheath, this fatty material contained within the Schwann cell itself. The Schwann cell has its own nucleus. Along the axon, you wouldn't find the nucleus, that's back in the cell body, but as we'll see in a minute, you would find mitochondria along here, but it's basically just cytoplasm or cell liquid with a bunch of ions dissolved in it. And then what you notice is where one Schwann cell ends, there's a little gap before the next Schwann cell wrapping around the axon begins, and that gap between Schwann cells is called a node of Ranvier. So again, you've got the tube of the axon itself filled with cell liquid or cytoplasm and ions, surrounded by the Schwann cells, which have their own nucleus and are filled with this myelin fatty material that surrounds and insulates or isolates the axon. And then between successive Schwann cells, you have a gap called the node of Ranvier. Now let's take a look at what happens when we put that, when we go down the length of the axon, and we arrive at its terminal end, which in the case of a motor neuron would be at a skeletal or striated muscle, and the axon branches out into what we call the axonal terminus or termini, a few different branches, that each of which then synapses or connects to an individual skeletal muscle cell. Those points of synapse or contact we call a neuromuscular junction, and the very end of the axon is called the motor end plate, where it forms that neuromuscular junction or synapse with the cells of the skeletal or striated muscle. I want to remind you that this is a very schematic drawing, and what's especially out of scale is the size of the axon. Remember that these are very unusual cells in that the cell body of the motor neuron would be in the spinal cord, and this axon could stretch all the way out to a muscle that might be out towards the end of your arm or leg, upper limb or lower limb, and so the axon itself might be almost a meter in length in some cases, but in cross-section it's extremely small, in fact so tiny that you can barely see it in a light microscope. So we've greatly exaggerated the width of the axon and sort of minimized the length just for the purposes of our drawing. And remember, that cross-section would be so tiny that what we're looking at here would look as just a tiny dot in the microscope, in the highest power on a light microscope. Now, what's so unique about these axons, and the reason that I've kind of made it as big as it is that they're able to create a voltage potential across the axon cell membrane. This is not electricity moving down the axon. This is just a difference in the ions that are inside the axon and outside the axon, creating a voltage difference. So let's look at this a little closer up, and what we're going to see and what's happening here at the axon cell membrane is there are these proteins embedded all along the axon membrane, especially in the nodes of Ranvier between adjacent Schwann cells, and these proteins have a special name. It's really a whole complex of proteins called the sodium-potassium pump, and as the name suggests, a pump moves things or pumps things, and in this case what they do is they pump ions across the cell membrane. positive ions. It's a little bit more complex story in terms of the exchange between sodium and potassium ions, but for our purposes, let's just consider that they push the positive ions outside the cell, leaving a net surplus of negative ions inside the cell and positive ions outside the cell, such that if we were to put a voltmeter on here, and this is possible with tiny glass electrodes, that voltmeter would register inside the cell a minus 70 millivolt potential compared to the environment outside the cell. We call that the resting potential. The reason we call it the resting potential is that the neuron is at rest. There's no impulse or action potential moving along the axon. It's just moved those positive ions outside the cell. This does not, however, mean that this is not an active process for the cell. If these protein pumps are pushing ions out of the cell against the gradient, they must use ATP to do that, and that ATP for the sodium-potassium pumps is going to be provided by mitochondria, and we would find mitochondria interspersed all along the axon to provide the ATP for this action. In fact, about a third of all the calories that you eat every day are used to create resting potentials in all of your neurons and muscle cells throughout your body. So it's a very active metabolic process. All right. In summary, what we have is a motor neuron with its cell body that would be sitting in the spinal cord where the nucleus is located, dendrites, which would be providing input to initiate an action potential if it's going to happen. We have that axon extending out to the skeletal muscle where those motor end plates synapse at the neuromuscular junction with individual muscle cells in the skeletal muscle. And remember, along the length of the axon, it's covered by these Schwann cells, which, when we look, have gaps between them that are called the node of Ranvier, the Schwann cells filled with myelin. And what happens at the nodes of Ranvier is that the sodium-potassium pumps are pushing positive ions outside of the cell to create an internal negative resting potential of about minus 70 millivolts, more negative ions inside the cell than outside the cell. What this does is has this cell ready, although it's at rest, it's ready at split seconds notice from an input through the dendrites to send an action potential down the axon and trigger the skeletal muscle to contract. That will be a subject for another video. That then is the motor neuron establishing a resting potential in its axon from big picture anatomy.